Okay guys, are you ready for part 2 of the Cycle of Life Lab? I'm so excited to show you what's happened over the course of a couple of days. So these are your tubes, and these are in the light. You can see the light sources. I have them on both sides. All right, so let's grab y'all's tube so we can look at it. Look at this beauty. Look at that. Can you not see how amazing? Look at that color. I'm gonna even bring it closer so you can see. Look at those little bubbles. They're little tiny bubbles if you look closely on the leaves. Little bit of photosynthesis going on, right? Isn't that beautiful? Okay, so look at that color, because you need to think back to what did this tube look like before we put it in the light. So on your data table, go ahead and record for the next day, day 2, part 2, record the color. And that should tell you whether or not there's carbon dioxide in the tube, right? So you can kind of think about what happened. So to think, because this is kind of a difficult lab, you need to think about the fact that this is a photosynthetic organism that you put in this tube, and this is the one that you blew with the straw. Okay, so be thinking about that. And now this tube a couple days later is blue, so that bromothymol blue in there. Remember, bromothymol blue turns yellow or green in the presence of an acid. In the presence of an acid would be carbon dioxide. So that means now it's turning back blue, which means it's not acidic. Think about why. ... Okay? All right, let's go get the tube out of the dark and see what that one looks like. Okay, here's the tube from the dark cabinet. Now really look at the bottom, because that's where most of what's happening is occurring. Okay, so this one, remember, had the bromothymol blue in it, and it was like dark blue from the beginning. Look what's happening now. You can see there at the bottom. But to show you a real comparison, I'm going to show you this tube from the dark compared to the one that was in the light. There you go. Look at that comparison, guys. So the one that you see that's on the left, that's got that tint of like a light yellowish green, that's the plant that was in the dark cabinet, not exposed to light. And the one on the right was exposed to light. Okay, so knowing what you know about these guys and the two processes of cellular respiration as well as photosynthesis, I want you guys to answer the analysis questions that go along with this lab. Okay, so I would just tell you it's kind of a hard lab because you have to think about it, but you can't just think about photosynthesis. You have to think about cellular respiration as well. So when you go to find the answers, look at your notes for both cell respiration and photosynthesis, and think about what's happening with the color change based on bromothymol blue in the presence or absence of carbon dioxide. Okay?